Hello, it's Mr. Singleton, one of the Associate Directors for Outward Academy Trusts. This lesson is culturing microorganisms. Now, this is just for triple award students or any student just doing biology. So if you're doing combined science, you don't need this part and you can go to the next lesson. If you are doing triple, then you need this lesson. And it's all about bacteria, growing them aseptically. So at the bottom here, there is required practical. So again, you need to know the method, you need to know the risks, etc. So we're going to look at how you can grow just one single type of bacteria. And then what you would do is look at how you can work out if certain antibiotics can kill that bacteria. So pre-starter on this, I just want you to think about eukaryotic cells and prokaryotic cells and how they are different to each other. I'll give you 2 minutes. This can be done on a whiteboard in the back of your book. Off you go. Things you could have had. In a prokaryotic cell, you do not have a nucleus. There is no membrane-bound organelle, so that includes mitochondria and chloroplasts. And generally, the prokaryotic cells are smaller, and I'd let you they have a smaller ribosome as well, because their ribosomes are generally smaller. Other things you may have put, they don't have a cell wall made out of cellulose. Only a plant can have a cell wall made out of cellulose. Today's lesson is culturing microorganisms. In your books, if you're using books, title, culturing microorganisms with today's date. If you are not using the book, you're using a revision card. This is still biology paper 1. And so put that on the top of your revision card and then the title, culturing microorganisms. I'll give you 30 seconds. The challenge for today is being able to calculate the zone of what we call inhibition of different antibiotics. And the aspire is to evaluate the effectiveness of the different antibiotics. So there's going to be a calculation in here, and we're also going to look at how we can look to see how antibiotics are effective. So there's a bit of a recap on prokaryotic cells and how they reproduce. So they reproduce by this thing called binary fission. And be careful with that one, because AQA come up with it a lot, how to prokaryotic cells or bacteria cells reproduce, and you need to put binary fission. What happens is the circular DNA and plasmids will replicate. The cell gets bigger and bigger, and just a little bit like mitosis, then the DNA moves to opposite poles of the cell, and then they divide to form 2 daughter cells, which are genetically identical. Now, the difference with this is, as you can see on this picture, the 2 bottom cells can have more DNA than one, or more plasmids than one, or less plasmids than the other. So the first stage up there at the top, the DNA duplicates, then it kind of goes to the middle, and then it gets pulled to the outside, and then they end up as 2 different cells. Oh, sorry, two new cells, but they have the same DNA and just different numbers of plasmids. So bacteria will reproduce very, very quickly when given the right conditions. Around about 40 degrees Celsius. If you're looking at this animation now, you can see that's a one bacteria cell reproducing. So sometimes about every 20 minutes it can replicate, which seems like it wouldn't be too bad. But if you think about, use by dates on food, if you think about the fact that, if you had one bacteria on there after 24 hours, that would be quite a lot. So let's see if we can do some type of calculation to work. So if a bacteria cell divides, we put this to half an hour or 30 minutes, how many cells will it have produced in 2.5 hours? So I want you to have a go at that, see if you can work it out. I'd start by thinking about how many divisions it's going to go through, and then think about how many times each of those cells will divide. Because that one cell will become 2, but those 2 cells will divide to make the 4, then those 4 will go to 8, etc., etc. Okay, I'll give you a minute. It's quite a hard one. Off you go. Right, let's see what. So this is how you would do it. 2.5 hours is 150 minutes. You need to convert it into the right time frame. So we've had hours, but you're talking about how many times it divides in 30 minutes. So that would be 5 divisions. So that's how many times it can divide. And then you multiply 2 by itself for the number of divisions. So it was 5 divisions. So it's 2 to the power of 5. And if you try and do that on a calculator, you usually end up going wrong. So you should be able to put to the power of and then put in a new number. It's usually on the calculator, it's usually an X, a big X, and then like a superscript Y. And then you can put whatever number you want in. So that would be 2 times 2 times 2 times 2 times 2, which is 32 cells in 2.5 hours. So what we try and do, if we want to look at, let's say there's a new strain of bacteria, and we want to see if we can kill that bacteria with a new antibiotic or with certain antibiotics. What we've got to do is grow that bacteria really quickly, and we only want to grow that type of bacteria. We don't want to grow lots of different types of bacteria, so we have to keep it, make sure there's no contamination in there. You've got to just grow the bacteria you are trying to test against. So we're going to grow one type of bacteria, and then we can put different antibiotics on it or different antiseptics on it and see if it kills the bacteria. And that is an aseptic technique is the way of working which stops contamination of your sample. So I want you to have a go at this Google quiz which I've set up for you. There's only a couple of questions in there. I'll give you 3 minutes because you might need to open it up, etc. So you've got 3 minutes. Have a go at the Google quiz. It recaps some of this. There's quite a big calculation in there. So please have a calculator ready. 3 minutes, off you go. Okay, you've got 1 more minute.
So this
is a required practical actually that you need to be aware of. It's the triple only. One of the triple only required practicals
studying biology by itself,
about how to grow bacteria.
And it's called aseptic techniques. It's about growing bacteria and just one certain type of bacteria, and then being able to see if certain things will kill that bacteria. So let's say I want to find out if this hand sanitizer here kills a certain type of bacteria, then what I have to do is I need to grow that bacteria very quickly and then put the hand sanitizer on it to see if it kills it. In order to do that, you need this thing, which is called an agar gel and an agar plate. AQA like you to know how to make this agar plate. So what it is it comes in a little broth, so it comes in a liquid, and what the technicians will do is they will sterilize the lid of that jar that they're using, and I'll show you how to do that soon. So they'll sterilize it, and they'll sterilize this petri dish, and then they'll pour it in and leave it to set, and then you get this nice agar jelly. Now this agar is like the food for bacteria. The bacteria love this stuff, so they'll grow really quickly on it as long as they're in a nice temperature. Okay, so that's an agar plate. It's been pre-prepared for me, but you should know how to make it. You pour the agar broth into a sterilized petri dish, and then you leave it to set with the lid on. Now, you may have done this previously in a different era, where you've gone around the room and you've got a cotton swab and you've tried to grow bacteria that's around the room on agar plates. Now, this is different. I only want to grow one type of bacteria. So I'm only going to be growing this, which is E. coli here. So this is a broth of E. coli. Don't worry, it has been changed so it won't hurt me. Okay, it won't harm me, this one. So it's been changed, and this is all I want to grow. Let's say we want to try and find out what kills the coronavirus. If we grow loads of coronavirus on our agar plates, wouldn't quite work like that, but a bacteria better, then we can put different things on it and we'll see if it kills it. So we can spray Dettol on and see if Dettol kills it. And that's what this is about, growing one type of bacteria and seeing what works against it. This is an important piece of equipment. It may just look like a piece of metal with a wire loop on the end, and that's exactly what it is. But in science, we call this an inoculating loop. And this is going to transfer my bacteria from the broth onto this agar plate here. Okay, and you'll also notice I've got a Bunsen burner there, and that's to help sterilize. So, I know bacteria is microscopic, so there could be bacteria all around this lid here, and there's almost certainly some bacteria that we don't want growing that isn't E. coli on this inoculating loop here. So the first thing I've got to do is sterilize this inoculating loop, and I need to sterilize the top of this lid here. Okay, so in order to do that, I need my Bunsen burner. This isn't easy to do with one person, so forgive me if some of this doesn't quite work. I'm going to turn this onto a roaring frame. I know you probably can't see that through the camera, but it's on. It's just very, very hot now. So in order to sterilize this lid, and this is what I mean about flaming a lid, I've probably got bacteria all around here that's not E. coli that I want to get rid of. So all I do is I just move this lid right around the flame, not for too long, but just to kill it all. And I'm also going to dip my inoculating loop into this ethanol. Now you see this big massive sign here saying it's flammable. You've got to keep this Bunsen burner away from this ethanol. Okay, so we keep that well away from the naked flame. That's part of your risk assessment. So my inoculating loop goes in here, and then it will go into my flame, and that will sterilize this inoculating loop. And I've sterilized the top of this lid of my broth. Now I've finished using my ethanol. That needs to go very far away, and I need to turn my Bunsen burner off completely, so I'm not going to get any issues with that. Okay, now, so here's my agar plate. What I don't want to do is open this right up, because I'm going to subject it all to the bacteria that is going to be in the air. So I just lift this lid up very slightly, okay? The less you open it up, the less contamination you're going to have. Then I dip my inoculating loop, which has been sterilized, into my E. coli broth and move it around to make sure I got some. Now, some of that E. coli is transferred onto my inoculating loop. I then need somebody to put the lid on that. But what I'm going to do here, I'm going to try and do it this way. I'm going to smear this inoculating loop with the E. coli on, and I'm going to close it, and I'm going to do a turn, and then I'm going to smear it across again. So we turn it, and this just makes sure we get a really nice spread of bacteria right across the agar plate. So one more, and then that should be spreaded really well. You can use this thing as well, which helps to spread. This is a glass rod that's been bent. It's called a spreader. Okay, so you can just smear that across as long as you've sterilized it. Make sure this lid goes on, because you don't want E. coli going everywhere. So my lid's been kept quite close. I've stopped contamination. I've spreaded the bacteria this way, and then I've turned it and spreaded it this way, and I've spread it this way. And I've made sure, to the best of my ability, I've only grown the E. coli which is in here. And I've done that by sterilizing it in this flame and also flaming the lid of the bottle, and that will stop any other type of bacteria getting in there. Not quite finished yet. I don't want this lid to fall off. So what I'm going to do is I'm going to tape it up. All right, because when this gets lots of bacteria on, we don't want lots of bacteria flying around everywhere, even if it is safe. So I'm going to tape my lid. Now, that is it. That is how you tape the lid. You do not tape all the way around, because you will stop oxygen getting into that bacteria. Okay, so you just put 2 pieces of tape on, and then you are going to need to label it. So I will put my initials. I know what's growing in there, and so then everyone else knows in there that it's E. coli, and then I can put the date, so I know when I'm going to take it out. And what I mean by take it out is it's got the right food, but it's not got the right temperature. So what we're going to do is we're going to put that into an incubator, which will keep at 25 degrees Celsius. Now, 25 degrees Celsius in a school, in industry, or when bacteria grow best, you're talking 40 degrees Celsius, but we don't want to grow anything too dangerous. So in schools, we only grow them at 25 degrees Celsius. It stops harmful bacteria growing. And that is how you grow your your E. coli on an agar plate without growing anything else.
So what I want you to do is, after listening to that, just the actions that I've carried out. So the inoculating loop is heated. Why do I do that? Which one does that match up to? The loop is allowed to cool before putting it into the bacteria culture. Why would I want to do that? The lid of the Petri dish is only partly open. Why do I do that? And then the bacteria is incubated at no higher than 25 degrees Celsius. I just want you to have a look at those reasons and match them up. If you want to do this very quickly, you're on a whiteboard, then just draw 4 boxes for the actions, 4 boxes for reasons, and just match them up. And if you've been a bit more conscientious, then write them out so you know which way they're happening. I'll give you 4 minutes to do that because it's quite a long task. Off you go. Okay, you've got 1 more minute left. Right, so the inoculating loop is heated on a Bunsen burner flame, and that's to sterilize it or kill any bacteria. The loop is allowed to cool before putting it into the bacteria, because if it's too hot, then obviously it would actually kill the bacteria. The lid of the petri dish is only partly open, and that's to stop any unwanted bacteria. Remember, we're only trying to grow, in this case, E. coli. We didn't want to grow anything else that might have been in the air, so it's only partly open to prevent any unwanted bacteria. And then it's incubated no higher than 25 degrees Celsius, and that's to prevent the growth of dangerous or harmful pathogens. Okay, now this is an exam question that's been set and been in the past. So 5 disinfectants were put onto this agar plate, and it wants to know which. The one the student said that D is definitely the best for using around the home. And most people usually get this one. Why might they be correct? But what they don't think about is the extra bits on this. Why might they be incorrect? So generally, most people got the first part of this question right, but then did not answer the last part right. So I'll give you a little bit of time to think about it. Why is D the best? Have a look at it. So if you see these petri dishes, where the disinfectant's being put, that's the innermost circle. And then all the shaded bit is like where all the bacteria is grown. And then where there is no area, so where it's quite clear or quite white, that's showing that no bacteria is grown there. Let's have a look at the answers. It might be correct because the largest area where no bacteria are growing or it's killed the most bacteria. So that's why we would expect that conclusion to be right. There was no bacteria in the largest amount of area, so therefore it's killed the most bacteria. However, the harder part on this is why it might not be, and that would be because it might be harmful to humans, or it might be harmful to animals, or it might like absolutely destroy your surface, which would be pointless then, wouldn't it? The other reason is we've only tested it on one bacteria, and it might work differently with different bacteria. So it's just about thinking of on the flip side of that coin, why else might it not be as good? Because it looked really good, but you've got to be able to come up with reasons why it might not be good around the house. And there was a clue there because it said around the house. So then you've got to start thinking about why have they put that in that question, and how can I use that to answer my question? So I want you to have a go at the Google quiz that I've set up for you there and answer some of those questions. I'll give you 3 minutes, off you go. Okay, you've got 1 more minute. We have completed the challenge, which is calculating the zone of inhibition. That's using the area of a circle. So that's how you calculate that area around where there is nothing growing. And we have evaluated the effectiveness of different antibiotics by looking at those slides, those bits where which antibiotic has killed the most areas. That was the exam question. This is if you would like to make the revision card. So if you're making a revision card, then please put biology paper 1 on the top of that that revision card. And then I would put growing microorganisms or aseptic techniques. Growing one type of bacteria is really important, so that means we can test new antibiotics to see if they work properly. Firstly, what you do is you make the agar plate, and you would get the petri dish, make sure it's sterilized, so you could rub some sterilizing fluid on there, then flame the lid of the agar, and that sterilizes it, and pour the agar into the petri dish and allow it to set. The agar, remember, are the nutrients that the bacteria need to grow quickly. Next, get the broth of the bacteria that you want to grow. In this case, it was E. coli that we used. Flame the lid of that just to make sure we're not cross-contaminating and then we've got any other bacteria on there. And flame the inoculating loop. That was that piece of wire. And that again will kill any bacteria that might have been on there previously. Put the inoculating loop into the broth and then part open the lid. Don't open it fully of the petri dish and spread the loop all over the agar evenly. Partially tape your petri dish and place it at 25 degrees Celsius in schools, and that stops harmful bacteria growing. It's such a common question, that one. Why do they grow at 25 degrees Celsius? It's to stop harmful bacteria growing. I'll give you 3 minutes to write that down. Again, if you're struggling, obviously just pause the video and then we will move on to the exam questions. Okay, you've got 1 more minute. We have come to the end of the lessons. I've made you some exam questions for you to have a go at. They're Google Doc, so you can either write on those or you can print them off and then answer them and put them into your book and hand them to your teacher next time you see them. But please do have a go at them. Well done, guys. Keep it up.